Okay, testing to see if the sound is through. Oh, yeah, good. Okay, all yours. No, usually you can just type a number. Sorry. There you go. All right. And sorry, you can do this. You can go to slide. Oh, yeah, sorry, that's Google Slides. I'm not very familiar with that. Okay. Sorry. You think it works? Ooh, that's sort of. Okay. Make sure that we're on the correct network. I just want to make sure that the network that we're using is MIT, because that'll go. Data center is like 4 floors up. So is that the one you wanted? No, it's a little farther, but just 1 second. Let me move this. That. There you go. All right, great. So last day we talked about this incredible algorithm, AlphaFold 2, and I just gonna go over that again and remind ourselves where we were. So this was a deep learning algorithm that learned to predict protein structures, and internally we had 2 representations, one from the multiple sequence alignment, and that carried a lot of evolutionary information with it. And the other internal representation was a pair representation, and that stored information about the geometry of different pairs of residues or different pairs of atoms. And we saw that algorithm worked pretty well and could learn to reproduce protein structures. So in addition to protein structures, and if you recall, one of the things that was really crucial for AlphaFold working was that the problem that it was optimized on was not just producing a final structure at the end, but there were intermediate objective functions such as being able to predict the distance or distribution of distances between any 2 given residues, being able to predict their orientation and the rotation around their phi-psi angles. These were all intermediate objective functions that really serve to embed geometric information into the pair representation matrix. The other important thing that they were able to predict as output to the final model was a predicted local distance different test. And so the LDDT really measures in a local sense how correct the structure of the protein is. And they were able to show that the predictions from the model were more confident in the regions that it had a better predicted LDDT. So the reason that's really useful is if you have a model that you know isn't perfect, but it can tell you whether or not it's working, then you can basically just keep refining the model, rerunning the model even until you get a score that's really good. So that's really one of the key features of a sophisticated model like this. So in general, so you can use these PLDT scores to tell if the structure that you're trying to predict, if it can be trusted, or if parts of this structure can be trusted or not. Generally, under a score of under 50 is not reliable. Above 90 is quite good, and you can use it for molecular modeling. And above 70, you can kind of assume the backbone structure is correct, but maybe not the side chains. Okay, so that was AlphaFold 2. More recently, AlphaFold 3 has come out, and I think there's a few interesting takeaways. So one of the things that I learned when I was diving into the AlphaFold 2 architecture is that I was very familiar with transformers. Hopefully, by the end of today's lecture, you guys will be very familiar with transformers as well. And in AlphaFold 2, they really modified the traditional transformer quite a bit to suit their problem. And really the organizing principle is thinking about which parts of the underlying representation need to share information with each other to be able to solve the problem at hand. And so in AlphaFold 2, there was a lot of information being passed from the evolutionary MSA representation to the pair representation and back. In AlphaFold 3, the architecture is greatly simplified. And one of the things that ended up working a lot better is the information from the MSA is really bottlenecked into the pair representation matrix. And the pair representation matrix ends up being kind of where all of the important information gets stored. And if you recall, that's where the geometric constraint comes in. So that ended up working quite well. And the other thing that's notable is, in AlphaFold 1, they used energy functions to predict the final protein structure. In AlphaFold 2, they had a neural net. And in AlphaFold 3, they have a diffusion model. And we'll talk a little, very briefly, about other diffusion models today. But it's really emerging as one of the more powerful tools in generative AI. But because they were able to kind of put all of the relevant information in this pair representation matrix, they were able to put information from other types of atoms in. And so AlphaFold 3 really integrates into protein structure, protein-protein interactions, protein-DNA interactions, and even interactions between proteins and ligands. And so, I think this is going to have a big impact on how it's used, because this really gets to, how do you go from a structure of a protein on its own to understanding how that protein is going to interact with other proteins or with DNA. So in AlphaFold 3, that's all combined and modeled in a single framework, and it seems to work quite well. So you guys will have some fun playing with AlphaFold on your P set if you haven't done that already. And a lot of these tools kind of bring us to like one of the other areas in structural biology that's really been impacted by AI and generative AI in particular. So protein design is now one of the big challenges. If we say, AlphaFold has solved the protein folding problem, now one of the big challenges is, well, how do we design our own novel proteins to carry out new functions? And those functions might be, hey, we want a protein that has an enzyme activity that we don't see in nature, okay? And how would we do that? Well, we need to think about the transition state, and there's quantum mechanical calculations that can be done with specialized software that says, oh, if you put your molecule in here and put just the right strain on it like this, this might catalyze the reaction you're looking for, and now we can go and design entire proteins that can try to stabilize that transition state of a particular chemical reaction, even one that doesn't occur naturally. We can design protein-protein binding. So if we have a gene that we want to target, we want to activate it or repress it, we can design a protein specifically to do that. We could design assemblages of proteins that maybe make a molecular cage around a molecule that we want to deliver, and with the right stimulus, they would open up. Biosensors are a big target. So can we design a protein that binds to some small molecule and then does something? Maybe it turns on a transcription factor or something like that, and gives off some signal. So these design proteins, they can have novel functions. They can have novel folds. So the protein that you see there, this was designed, I think it was the first protein design that had a novel fold. And by fold, it's sort of the sequence of alpha helices and beta sheets and the way they're connected together. This had never been seen in nature, and this was actually designed while I was a graduate student in David Baker's lab. The protein is called top7. I think it has something to do with the topology. It has seven it has 7 units of secondary structure, although it might have been the 7th iteration. I don't really know where the 7 comes from. But that particular sequence of beta strands and alpha helices was not seen in nature, and so this was really a test. Can we design new folds, or do the folds we see in nature, are they special for some reason? The other interesting fact about this protein is I know my friend Gautam Dantas, who was one of the graduate students who was working on this project. When he went to purify it, we would always purify these things by, first unfolding them in, really high concentrations of guanidine. And what he noticed is that, he got up to like about the solubility limit of guanidine, and this thing wasn't unfolding. And so these design proteins can actually have stability that's much greater than that of evolved proteins. Well, why is that? Well, once you're stabilized by, let's say, 10 or 15 kilojoules per mole, you're going to be folded like 99.999% of the time. So there's no pressure in evolution to, go from that to like 99.99999 repeating, right? So because that's very difficult to select for, we can actually design these proteins that are like, really rock solid. Okay, so when I was in graduate school and they were working on that, there was a lot of intervention and a lot of trial and error to generate that first design protein with a novel fold. Now we're getting closer to workflow, and pretty much every stage of this workflow is really enabled by a lot of these new deep learning tools. So what does an example workflow look like? Well, we start by generating a backbone. So we have to figure out, what is the shape of the protein that we want, and that's going to be most likely informed by the function that we want it to have. And so that could be a rational design, or we can come up with a concept and then use a program called RF diffusion to generate examples of protein structures. Once we have an idea of what the structure should be, then there's additional software like protein MPNN that can basically guess a sequence given a structure. Then we build that sequence and turn it into a structure using AlphaFold. And if that works and we get a good PLDDT, then that might be worth trying to make in the laboratory. Otherwise, we can go back and we can slightly modify that original design by pulling some neighbors out of RF diffusion, or maybe we just go back to the protein MPNN step where we're guessing the sequence, and so we can iterate this at kind of any step, and that's still ongoing research in terms of what's the best way to do that. Okay, so I mentioned diffusion models a couple of times. We're not going to talk about them today. I don't know if we're going to talk about them later. It'll really depend if I find any really cool diffusion models in the next unit on drug discovery. But I will say these are really emerging as kind of one of the most popular tools. And you're probably familiar with stable diffusion and some of the diffusion models that generate images, and sort of the very high level intuition on how they work is these are models that have neural nets that are trained to undo the noising process that you're looking at here. So on the left, you see this picture, relatively clear picture of a dog, and on the right, you see complete noise. And so we know how to go from left to right. We know how to add noise to this system. And basically, the idea is to train a neural net to remove noise from the system. And so we can do this with images, and really all of the I think top generative AI tools for making high quality images or high quality videos are based on this diffusion model. But we can also use that for protein structures. And so here you kind of see the same process where they've just taken all the atoms in the protein and they've moved them around. They've added Gaussian noise to the positions of those atoms, and then they've also added noise to the rotations. Okay, and then they learn a model that helps to undo that, and that allows them to. You can put new noise into the model. It's very easy to generate noise. We know how to do that. And so you can start from a different starting point and denoise it into a different protein structure. And so the hard problem is saying, oh, here's a bunch of protein structures. How do we sample from this space? So we take the protein structures, noise them, create a completely noisy signal, and then say, okay, we know how to sample from random uncorrelated noise. Now we're just going to reverse that process, and so we can take many samples in that noisy configuration, and then kind of work backwards through these physics-inspired tools to generate protein structures. Okay, so that's RF diffusion. It gives us some ideas for what a protein structure might look like. Protein MPNN is worth talking about, although we won't have time to get into it today. But basically, it has a structure that we're going to see again and again, and is repeated in transformers, where we have an encoder and we have a decoder. And our encoder is going to take in one data type, and that data type is a protein structure. And then it's going to decode it into a sequence. Okay. And in general, there are many types of encoders and decoders. We can have autoencoders, which will take in a data type and then in an encoder, and then decode it and try to reproduce the data that was put in. But this is a very common structure. Okay, so what's the state of the art in protein design? Well, there's a lot of success in designing proteins that bind other proteins or bind small molecules. I already showed you examples where we can use protein design to design novel protein folds. There's been impressive work designing quaternary interactions, so complexes of proteins that fit together. And we're moving toward the horizons of identifying proteins with multiple conformations or different enzyme activities, or really designing in different dynamics in these proteins. Okay, so that's protein structure. I want to move back now to protein sequence. And so on Thursday, you guys will learn all about protein language models. And that's really, these are models that operate in sequence space. So protein language models, or PLMs, are like LLMs. They're like large language models like GPT. But they're designed to model the language of proteins rather than natural human language. Protein language models in general are quite large. And so that means they're fairly cost prohibitive to build yourself. But there are very good models out there that you can use, and you can also fine tune on a smaller set of sequences. But in general, we can use these protein language models. We can look at the embeddings of our proteins in those models, and then we can use that for other tasks. And that's an example of transfer learning. And on the PSET, you guys are going to do that. You're going to take a protein sequence. You're going to pull out the embeddings. So you're going to look at how that sequence looks like in the final layers of the neural net of, I think, the ESM protein language model. And then you're going to use those embeddings to predict the subcellular location of the protein. And the nice thing about that is when you do that, you add a lot more information to the sequence. So you start out with, if you have 100 residue protein, you start out with 100 integers. But once you pass it through the protein language model and then you pull out the embeddings of the final layer there, you'll have hundreds of thousands of numbers that describe that protein, and there's a lot of information there, and so it will be easier to predict to perform your prediction task with a smaller number of examples to train on. Okay, so these protein language models and large language models are both based on the transformer architecture. And so for most of today's lecture, I'm going to go through in detail and give you guys a lot of information on the transformer. And I hope you'll find it useful. You're not going to have to follow all of that to be able to do the PSET. But I'd like to go through it in detail because, just like it's really the basis for, I think, a lot of different algorithms once you get the intuition behind it. I think you guys will have fun designing your own transformer-like architectures if you're working on a unique problem that's, let's say, a little different than sequence modeling. Okay, so to understand the transformer, let's go back a few years before, I think 2017 is when the main paper describing transformers was first published. And let's look at what were people doing at that time. And so if you guys remember, if you used Google Translate at all before about 2018. It was based on something called a recurrent neural net. And so the problem here is we have a sequence of text. So in this case, you were doing English to French translation, and the input is the cat ate the mouse. And the output should be the same sentence, but translated into French, right? And so in this case, we don't really know what is the length of the sequence a priori, and the length of the output sequence doesn't have to be the same as the length of the input sequence, okay? And so what would happen is we would feed in each of those words independently, the cat ate the mouse, and we'd allow the model to perform some sort of computation. And at every step, the model and the parameters of the model are the same. So it's the same model that's acting on the word the, and the same model that's acting on the word cat. And it's just being called recursively over and over with different data, and then there's a lot of logic on how to pass forward, parts of the previous computation. Okay. And so again, this is like that encoder-decoder architecture. And it's like a game between two players, right? Um, so I won't do this today because I think we're short on time, but in the past when I've given this lecture, I've asked someone to come to the board. I've whispered, like a movie title in their ear, and then had the class guess, like what movie are we talking about? And that's really so it's like a game, right? It's the first person is the encoder. You're taking this concept and you're converting it into a latent representation, which could be, writing on a chalkboard. And then everyone else is the decoder. You're taking that latent representation and you're trying to, figure out what was the data that was actually encoded. Um, and our decoder really is trying to um, pull out information that looks like it was drawn from something in our training distribution, right? And so this is really useful for obviously machine translation, image capturing, not capturing, captioning, things like that. And so if we look at each of these two parts, encoders, they do kind of one of two things usually. It's either dimensionality reduction, so we can take, a giant data set that might be difficult to understand just, purely by looking at the raw data. So if we have, let's say, a 2D image and a picture of a cat, um, we want to take all of those pixels with, like millions of numerical values, and then we want to reduce it to a much lower dimension object, and something, something that says like cat, or gray striped cat, or something like that. And that's great if we can, take all of our images, reduce them into this compressed space, and then, we'll need some other architecture to expand that out into an image. Okay. So one task is dimensionality reduction. So take complex data, collapse it down to a simpler form. Another possibility is dimensionality expansion. And this is what we're going to see in our protein language models, right? Because we take a bunch of integers, very small amount of data, but we're going to use what we've learned from, looking at hundreds of millions of other proteins, and then we're going to add more information, right? And hopefully it'll be high quality information to our input, which is quite small. And that information might allow us to make good guesses about properties of that sequence. And so an example would be ESM fold, which is like AlphaFold, but it takes a sequence in, puts it into the language model, adds a lot of additional information to that sequence, and then is able to, by virtue of the fact that it has that additional information, it can output a structure for the sequence. Okay. And we can do this in two different ways. If we have labels, so if we have a bunch of images and some of them are labeled cat and some are labeled dog and some are labeled plant, then we can structure our latent space so that we group those together, so we can do supervised training, or we can do unsupervised training and just put all of our data in and generate whatever latent space is kind of most natural for that data. Okay, so that's the encoder. Decoders are really kind of the workhorse of generative AI. So with our encoder, we can take in a picture and we could output the word cat or dog, or we can, perform some classification. The exciting, one of the most exciting things happening now in AI, of course, is generative AI. So we don't want to just like output the word cat. We want to like start with the word cat and then output new pictures of cats, right? And so that's really what the decoder is doing. And so if encoders are taking our complex data, putting it into maybe a more compact and meaningful latent space, our decoders are taking an object from that latent space and then decoding it into like a complicated object, like a 2D picture. And so the beauty of this is we can go into that latent space, and if we know where the we have an encoder, an autoencoder, so something that encodes and tries to decode to the same thing, and we put a cat in, we can go into the latent space and just move it around a little bit. Choose a point that's very close to where the original cat was, decode it, and hopefully it'll be another cat or something like that. So being able to sample points in the latent space and decode them is kind of the workhorse of generative AI. So we can use this for sequence generation, as we saw with protein MPNN. And we can also do something called conditional generation. So if we train our encoder in a supervised manner, where we have that label cat as we're encoding all those images, then we can generate images conditioned the same way. Okay, so this is the basic idea, and pre-2017, this is mostly how language translation was done with these encoder-decoders. And so the real issue here that led to the development of transformers is we might have a fairly long text. So for this particular sentence, very simple sentence, the cat ate the mouse, we can probably get like a pretty good translation of that. But as the text gets more complex and longer, we end up with this problem where we're just calling exactly the same network over and over and over for every word in our sequence. And then once we process that word and do some computation, we're gonna pass forward some information, but that information is it's like a fixed length vector, right? So it doesn't even if we put more information into the encoder, it all gets bottlenecked into a fixed length vector. Okay, so maybe that's 1,000 real numbers, but it's still probably not enough information as the thing that we want to encode gets more and more complex. Okay, so that's a basic idea, and then from that bottleneck, from that fixed length vector, or some combination of several fixed length vectors, we have to be able to decode it into a sentence or whatever it is we're trying to generate. And the idea is behind transformers is, well, what if while the decoder was working, what if it could go back and look at the input again? And while it was working, like, what if it could go back and look at the other words in the sentence that it had produced so far? If it could kind of like cheat and peek back at those data, it might have a better chance of doing what it needs to do. Okay, because in our encoder, whatever information goes in, it's like the telephone game. It has to get passed to the to the next instantiation in the sequence, and the next one and the next one. So by the time you get to the end of the sentence, it may have lost a lot of information from the beginning of the sentence, and it's just nice to be able to go back and look at it directly. Okay. So that was the idea behind attention. And so, even before the original transformer paper, there was another paper that described this attention mechanism whereby, as you were doing this translation, you could go back and you could, sneak a peek at the original sequence. But it was still, everything was still kind of bottlenecked through this recursive neural net. With transformers, so it's this famous paper called "Attention is All You Need." The reason it's called "Attention is All You Need" is this attention mechanism was developed, and it was kind of thrown on top of a recursive neural net. And what the authors here said is, well, gosh, you don't even need the recursive neural net. Just take every word, put it through your network in parallel, and just let all the words like see each other and share information between each other. And that's basically the concept. Okay, so that's what we're going to walk through today. This is a diagram that you'll see a lot when people are describing transformers. This is basically like the figure 1 from the Attention is All You Need paper. And I want to go through like all of these steps and point out what's going on, just so you guys like have some understanding of it. Okay, so this is an encoder-decoder model. So the encoder is basically that vertical column on the left-hand side where you see the inputs are going into it. Okay, so that's the encoder, and the output from the encoder is basically the output coming out of that last layer. Okay, and when you see that box and it says NX, that just means like that block is repeated NX times, I guess. Or, yeah, N times, I think is what that means. So you'd have any number of different numbers of repetitions of this basic unit. Okay, in each of those repetitions, so within each of those, that's a transformer block. It's going to have some parameters, and if there's like 8 transformer blocks, then they're all going to have different parameters. So they're all doing something different. So we can kind of interrogate what's going on in each of those layers, and it's going to give us a different view of like what's going on in that sequence. Okay. So on the left, we have the encoder. On the right, we have the decoder. And you can see that there's going to be information going into the decoder through in really 2 different ways. So first, you can see there's an arrow going from the encoder to this thing that says multi-head attention. So we're going to allow our decoder, and kind of if we go back here, the decoder is on the right there. So we're going to allow the different positions in the decoder to look at what's going on in the encoder. It's not going to look directly at the input that goes in. It's going to look at what the activations in those neurons are after the final block of transformers. So after the transformer has converted it to whatever that final representation is, that's going to be what the decoder gets to look at. Okay. And then the other piece of information that's going into the decoder is what you see at the bottom there, where it says outputs shifted right. And so initially, we're going to start it with a special token that says, "Oh, this is the first token in our sequence, or the first word in our sentence." And then once we use that decoder to predict the next word, we're going to take that next word, put it into the input, predict yet another word, and then we'll take both of those words, put them into the input. And so it's autoregressive process where, we keep predicting word after word, and that now becomes the input to the decoder. Okay. All right, so let's walk through first the encoder. And so there's a number of different things going on here. First, we have our inputs. And in this case, our input, if we're talking about words or sentences, our input's going to be tokens, like we think of those approximately like words, but we're going to convert those to integers and probably to some kind of one-hot vector. That's gonna be our input, and then we'll have an input embedding layer. So we talked about word to vec last time. That produced these really interesting diagrams where the name of the country was the name of the capital of a country was really predictable by taking the vector for the country and having some offset. It gave you the capital, or the offset from man to woman and king to queen was the same. So we remember there are these latent spaces that could represent the tokens and the words that we have in some more meaningful way. In our protein language model, probably not as rich as the embeddings for natural languages because we just have these 20 amino acids. But our inputs are first going to be go through this embedding process. And so they'll go from a single integer to basically a vector of 12, in the case of ESM2, 1,280 continuous valued numbers. Okay. From that input embedding, we're going to add something called a positional encoding. It's super confusing. And so I'm going to, I think I'm going to, actually, I don't remember where I put it here. It's super confusing. I don't want you guys to spend too much time on it because I think now there are better ways to do positional encoding. But what I want you guys to take away from this is what the transformer does is it takes all of our input tokens and it processes them in parallel. And we don't have to worry about the sequence. We don't have to worry about, oh, this one came first and then this one and then this one. We're going to do everything as if, the order of the words didn't matter. But we need to put some information into the model that gives it a hint at, like where those words or amino acids appear in the sequence. And so that's what the positional encoding does. It's actually just a vector that gets added to the semantic vector that that comes from looking at the amino acid alanine and then saying, okay, here's a here's a vector of size 1280 that I think represents alanine. We're just going to add another vector to that, right? And last time we talked about when we're operating in a 1280 dimensional space, we can add lots of stuff together and like all the information is still there, right? They don't really cancel each other out. So yeah.
Yeah, my question was when we try to update language transformers, like don't actually we don't require it quantitative, but you can do well without it, right? So
you can do well without what?
Without the positional encoding, and you just say.
No, there's so the original transformer paper was on language translation, and so that's where they came up with the positional encoding. You definitely need the positional encoding because otherwise, like the cat ate the mouse is the same as the mouse ate the cat, right?
How like something like attention figures out who did what and something, and so.
How does it do it? I'm so fascinated by that. I mean, that gets into I mean, there's the concept of like how it's planned and built into the architecture, but then like how these models actually work and how much sophistication is really in there. like these are like surprisingly open questions, right? Um, and I mean that all kind of gets into the field of mechanistic interpretability. It's like, what the heck are these things doing? Um, but I'll show you what the positional encoding looks like, and then if we have time at the very end, I'll show you some of the new alternatives to positional encoding that are becoming popular. I just say it up front because, like, when I read the paper the first time, I was like, what in the world is this? Okay, so we're going to add a vector to our embedding that says, like, hey, I'm residue number 7. And then we're going to take that, perform this operation that we'll call attention, okay? And then we'll get some new vector out, and then we're going to add it back to the first one. Okay, and this is called a residual connection, where we just add the original value back, and we'll talk about that later too. And then we're going to normalize it. Okay, and in the important thing that I want to point out for attention is that it looks kind of like a feed forward network, like a simple neural net. But attention does not have a nonlinear activation. So all attention does is it takes, if we think about our sequence, it starts with an embedding for our 20 amino acids, but then it gets modified, and it ends up having a lot more information. And we can think about this vector in this 1280 dimensional space. There's some direction of that vector that says I'm a hydrophobic residue, or I'm a positively charged residue, right? And because it's 1280 dimensions, there are other like directions there that say I am a positively charged residue surrounded by other hydrophobic residues. Or, I'm a hydrophobic residue that is preceded by a proline, or, just whatever features end up being important to be able to perform the task at hand, which is either predicting the sequence or predicting the structure. Those are going to be encoded. And so what attention does is it's able to look at each of those, let's call them features of the amino acid, and it's not able to do a nonlinearity. So it can't really transform them in a very interesting way. It just says, for this particular part of the task, I think the hydrophobicity is really important. And I think the fact it was, preceded by a proline or something like that is less important. And it's really just kind of like weighting all of these different components of that initial that latent vector that's going in. Okay. So it's not doing a computation in the same way that a neural network is. What it's doing is communication. It's sharing information across the different positions in the sequence before it does a computation later on. And then the next component of our transformer block is this feed-forward neural network. That's where the computation is happening, right? So in the previous block, like, it can't do interesting logical computations like exclusive OR, right? It doesn't have that nonlinearity, but each one of those is followed by like a computation block. So we think of that first part as communication. The second part is computation. And again, for both of the blocks, we're going to add the input back. Okay. Okay, so I'm just going to mention this just for completeness, This is a big problem, a very interesting and important problem if we're building a translation system. We have to take our sequences, break them down into words or something that, kind of approximates words. And we have to like actually come up with some infrastructure to take natural language and break it into tokens. And then the tokens are what we're going to feed into the model. For our purposes, we just have these 20 amino acids. Like we're almost certainly just going to use the amino acids as the tokens, and then we'll have a few special characters just to indicate like the start and end of the sequence. We're going to take those tokens, which are just integers, convert them into this initial embedding vector. Okay, and as I said, for ESM2, it's quite large, 1,280 real numbers. And so now we've taken our protein, let's say it's size 100, and now like to represent that even after just after the initial embedding step, it's now like 128K, right? It's a sizable thing. Now, if we were to take that 128K vector and try to do interesting downstream applications with it, like right after the embedding layer, I don't think we would get any performance that was any better than if we just took the sequence, right? Because we haven't added any information to it. But once we push it through these transformer blocks, the embedding that comes out is going to have a lot more information than the vector that we put in, right? And that information is coming from, in ESM2's case, the 650 million other protein sequences that it's been trained on in the past. Okay. All right. So I said I would talk about this positional embedding. I said it would be confusing. But it is true that order matters. So we want to add some positional data to our sequence. And so basically what they did in the original paper is which ends up working like fairly well, is they put in these sinusoidal position embeddings. And so they're basically vectors of sine and cosine of varying frequency as we move down the sequence. So this is kind of what it looks like if we were to plot them out. Okay. And so this would be for like a 50 residue protein. The vector that you see at the top, just like 1, 0, or 0, 1, 0, 1, 0, 1. This would be added to the embedding of the first amino acid in the sequence, and so on and so forth. And you can see it's a very different vector that's being added for the 50th. But what you can kind of see is most of the information, it's being stored in kind of the left-hand side there. So it's not using up that much of the space overall, right? So there is certainly a lot of additional information in there. And if we're training our embeddings at the same time that we're putting in these positional encodings, I would guess that a lot of the information that says, "Hey, tryptophan is very hydrophobic," well, it's probably going to be stored over there on the right-hand side where it's not really clashing with all of the positional information on the left. So these models will figure out how to efficiently use the space they have. So why do we have these crazy sinusoidal encodings? Because if we just do like a dot product or cosine similarity between different parts of the sequence, we end up with something that looks like this. And it says that residues or words that are near each other, so the I/J is close to the diagonal, those are considered very similar, and the farther apart I and J get, the less similar they are, right? So when we take a dot product between two of these vectors, they might have semantically, maybe they're both hydrophobic. They might have some association there. But then also in terms of their position, if they're really close to each other, they're going to have some association. Okay, so that's basically the idea behind the positional embeddings. Once we get past that, now we have these vectors in our transformer, and the architecture that I showed you at the beginning is sort of misleading because what's really happening is all of these tokens, all these words, all these amino acid residues, they're being processed in parallel. So they're all basically going through essentially in an identical version of this transformer block. But what they're allowed to do is share information with each other. And so you can see in this picture, we've got these four positions in the sequence, 1, 2, 3, 4, right? And you can see those like dotted blue lines, like position 4 is sharing information with position 3 and 2 and 1. And the attention mechanism is how we figure out what information they get to share. Okay, so there's our attention. Everything is connected to everything, but the weights of those connections are going to be very different. Okay. Okay, I'm going to mention this really briefly because I said we're going to run our attention, and then we're going to add the original input. So that original embedding vector goes through our attention mechanism where we get some information from other residues. We'll describe what that is in a minute. And then we're going to take the answer from that information sharing update, and we're going to add it back to the original. But in the diagram, it said add and norm. And so this was a really influential paper, the first paper on batch normalization. This was really important for being able to train, one of the things that was really important for being able to train very large, deep neural nets efficiently in a transformer. So basically what it is we just set the mean and the mean to 0 and the variance to 1, let's say for each particular feature in our input. And we would do that normalization over all of the inputs that we put in together as a batch. So let's say we have a batch of like 1,000 inputs. We look at the first feature and we just normalize it. So the mean is 0 and the standard dev is 1. That doesn't work for protein sequences because the first sequence might be 100 amino acids and the next sequence might be 1,000 amino acids. And so not all the features really line up very well. For these guys, we're just going to like look at each sequence on its own, look at all the different amino acids in the protein chain and say, okay, here I'm going to set the mean to 0 and the standard dev to 1 within the sequence. And so that's called layer norm. Do you guys know what I'm talking about when I say that the mean is 0 and the standard dev is 1? Like the mean of what? No, not the batch. The activations in the neural net. So when we have one layer of the neural net, those neurons are firing. They're sending out like whatever number it is, 0.2, -3, whatever. We're saying at that particular layer in the neural net, we want the activations to be normalized in some way, right? In general, for lots of models, we normalize it at that layer across the whole batch. For the layer norm, we just normalize it, across all the different copies of the transformer block that are kind of running in parallel there. Okay, so this is like the heart of the transformer. Okay, so you guys can you see the workflow. We'll go through it again at the end. It'll probably make more sense. This is the crux. Okay, this is where all the action happens. Okay, so we have these data vectors that are going in, and each one of these data vectors. So this this is our 1280 dimensional vector that currently represents position number 7, a bunch of numbers. We're going to convert it into a query. How are we going to do that? Well, we're just going to multiply it by a matrix, right? It's a vector. We're going to emphasize some parts of that vector, right? I can generate a matrix that will take that vector, figure out what is the direction that, the farther I move in that direction, it means this residue is hydrophobic, right? And I can really emphasize that in my query, right? I can say the query is all about how hydrophobic it is. We're going to do the same thing with matrix of keys, okay? And so that, that might also, it could be the same thing. It could be a matrix that says, it just measures how hydrophobic the amino acid is, or measures how hydrophobic the general vicinity of that amino acid is. Okay, these are this is what we were talking about before. We don't know how these things are working, but probably there's a lot of things like that, a lot of features like that going on in these models. And I think one of the most exciting areas is diving into these models and saying what are the features that are important. So our query might be how hydrophobic is this amino acid. Our key might be how hydrophobic is this amino acid. They might be very similar. And then if we take a dot product between those two, it's going to link up amino acids that are both hydrophobic, right? And it's going to say hey, these two amino acids should be sharing information. Now, on the other hand, I might have a query that points in the direction of this residue is positively charged. And then I want to say well, what other residues should it pay attention to? Well, then my key matrix might be just emphasizing a vector that points in the direction of this residue is negatively charged, right? So my queries and my keys might actually be very different, right? They don't have to be the same, and it makes sense in English language too, because the two words that are really important to understand together, it's really the same word, right? It's not a repetition of the same word. It's just another word that in this context, is really important for understanding the first word. Okay. Okay, so we have this matrix that emphasizes different parts of our amino acid representation. That is the amino acid we're querying. We're comparing it to this other matrix that's emphasizing different parts of a different residue. And we're going to multiply, basically take both of those residues. So let's say residue 7 and residue 10. And we want to know, does residue 7 interact with residue 10? So we're going to multiply our vector for 7 by our query matrix. And we'll get, we'll get some vector coming out. We'll multiply residue 10's representation by its key matrix. We'll get another vector that comes out. And then we'll take a dot product, and we'll say if these two vectors are aligned, then residue 7 and residue 10, this is important. Residue 10 has important information for residue 7. Okay. And we're going to step down for, if we're focused on residue 7, we're going to compare it to residue 1, 2, 3, 4, 5, 6, and then 8, 9, 10, all the way to the end of the protein chain. And we're going to compare the query for residue 7 with the keys for all of the other ones, right? That's going to give us a matrix that has the size of the sequence length by the sequence length, and each entry in there is going to be the dot product of the query and the key, right? And so if we look across the rows of that matrix, that's called the attention matrix. And it seems a lot like that pair representation matrix from AlphaFold, right? It's telling us which parts of the sequence are relevant for each other. So if we look across the 7th row, it's going to tell us how important each of the various different residues are for residue 7, okay? So now we need to figure out how much information to take from each of them, and we want the total amount of information that we take to be constant, right? So that's where we're going to use our softmax, right? Whenever we have some output from a model in terms of logits, and we want to convert it to a probability. In this case, we want the probability, for all of the different residues that aren't residue 7, we want all those probabilities to add up to 1. We know that's what softmax does, right? It just weights them by e to the minus logit, and then divides it by the denominator of the sum of all those things. Okay, so now we have this pair matrix, and what we're going to do is it's going to emit some information. And what information should it emit? Like if we're saying residue 7 and residue 10, we did that dot product of the query and key, and we decided that like they need to like share some information, and we're going to add that information back to residue 7. Like what's the vector that we should add back? It's not obvious. So we let the model learn it, right? Like we let the model learn this matrix for keys, queries, and values. Like the number that's getting added back to the vector for residue 7. It's just a matrix, and we're gonna optimize it and figure out what those parameters should be. Okay, so let's kind of walk through it. So we can imagine that X here is our input data. Okay, and so in this case, it looks like I have 2 rows. So let's say there's a sequence of just 2 residues, right? And the latent space, each one of those residues is represented as a 4-dimensional vector. Okay, so I have 2 residues, 4 dimensions. I'm going to multiply that by my query matrix, so WQ. And now I'm going to end up with a matrix that it still has 2 residues, but the dimensionality in my attention matrix is different. It's now a 3 vector instead of a 4 vector. Okay, and in general, like there's no reason that the dimensionality of the query has to be the same as the dimensionality of the input. Okay, so that becomes our a set of 2 query vectors. We're going to multiply it by our key vectors. Okay, and we'll do the transpose of the key. So it's really just a dot product. Okay, and then we'll end up with this square matrix, and it'll have 4 positions, and it'll say residue here's how residue 1 relates to itself in residue 2, and here's how residue 2 relates to itself in residue 1. And then we'll take that, and then we'll multiply, do our softmax, and then multiply that by the values, right? And now our values, like, it's still going to have the same dimensionality as our queries and keys, which is not the dimensionality that we started with. It's, here it's 3 instead of 4. In our case, our input X is, it's probably going to be like 100 rows for 100 amino acids, and then 1,280 columns, right? And our query key and value, it probably won't have 1,280 columns. It'll probably have less. Okay, so I think this is kind of redundant to what we just talked about. There's a rescaling that happens that honestly I don't. I think it's relatively straightforward. I don't remember off the top of my head like why it's the square root of the dimensionality. But that's as may be. We take that matrix, multiply it to get our values. That's kind of our overview. I wonder if I have, okay. So usually with attention, I said there could be attention where the queries ask, like, is this a hydrophobic residue? And then the keys are also asking, is this a hydrophobic residue? Whereas there could be other, like attention matrices that say, oh, this one is positively charged. And then the keys are like what's negatively charged. In general, it's probably going to be very difficult to build a model that can encode all of those things simultaneously. So when you see an attention layer, there's usually more than one attention head, and each attention head is just a separate query key value weight matrix. Okay? And in general, if those the dimensionality of that comes out of those weight matrices tends to be smaller, and then what we do is we just take all of the outputs from those different attention heads and we just concatenate them together to make a new vector. Okay, and so then if we have 8 of those 8 attention heads, then if we didn't make the dimensionality of the weight matrices like a little smaller than 1280, we'd end up with like an 8,000 dimensional concatenation that we would just have to downscale to 1280 anyway. And so there's another matrix that comes in, and it takes whatever the dimensionality is of all the different attention heads when they're concatenated together, and it just converts it back to 1280, and it does it in a way that hopefully keeps a lot of the information that was already there in the initial 1280 vector. And the reason we know this almost has to be true is, remember, we're taking that update that's coming from our attention mechanism, and we're just adding it to the thing that went in. So it's got a, even though we've taken the circuitous path and added all these numbers from other weight matrices with different dimensionality and concatenated them together in some arbitrary way, we need to put them back into a format where it makes sense to just add that to the initial one. And so that's what this additional matrix WO is doing, where O, I think, stands for output. Okay, so this is just being explicit about that. So each one of these steps, we are adding the thing that went into it. And as we just described, it helps remind us that there was already some structure to this vector, and so we don't want it to, be super crazy when we concatenate them, the output of the attention together. We want to, maintain some of that initial signal. And this also helps a lot in very deep neural networks, where, in order to train them, we use an algorithm called backpropagation. And backpropagation really looks at the derivative of the output with respect to each one of those parameters in the neural network. So for many, many layers away from the output, then that gradient can start to flatten out, and it kind of disappear. But if we keep adding the input to each of those transformations, it helps that signal persist a little longer on the network, and so it's a little easier to train. And this is so this was the initial paper that used these residual connections or skip connections, where we just add the initial vector to a later step. And what you can see is on the left, I don't even remember. Oh, it's image recognition. It was an image recognition task. And what they found was initially, the more layers they put in their network, in many cases, like the worse it would get. And so so you can see on the left, the 56 layer network has like 15% error. The 20 layer network only has a 10% error. And then once they added these residual connections, they were still getting improvement all the way up to 110 layers. So this really kind of helps the training process as much as anything else, and it allows you to go to networks with bigger scale and more parameters, and still be able to get something useful out of them. So that's kind of a motif that you'll see a lot in many different network architectures. Okay, I think since we're approaching the end, I do want to mention the decoder, but we've really been through all of the components now. So let's just take a look at the entire structure. So on the left, we have our encoder. Our encoder has, we take our input, we add our positional encoding, those weird, sinusoidal patterns, get this 1280 vector. That goes into a multi-head attention. So, let's say it's like 8 or 16 different query key value matrices that are independent from each other. The results of that get concatenated together, reduced back to our 1280 dimensionality, and then we add the original incoming vector in. No nonlinearity yet. And then we take that and we put it through a feed-forward network, where we do have a nonlinearity. We have like a ReLU gate or something like that. And then we add another one of these residual connections. Okay. And all the time we're normalizing. So our mean and standard dev is the same for all the different residues in our sequence. That's feeding into our decoder. So in our decoder, we would let's say start with a start token, or maybe like a sequence of different tokens, and we'll have some positional encoding that we add to that. We'll have another self-attention layer, And then we'll have something different. So in our encoder-decoder, we want the decoder to not only be able to look at like what are the other words in the sentence that I'm, producing in French, but we want it to be able to look back at the English sentence. And we're going to look at the output of the very last transformer block in the encoder. And so that's a cross attention. So we do a self attention, we do a cross attention, and then we put that into a feed forward network. And we do that like some number of times. And then at the very end, we'll have one more or maybe a couple more linear layers, just a really simple feed forward neural net. And then a softmax, and then we'll have our probabilities, whether it's probability for a particular word or an amino acid or whatever it is we're trying to produce. So just for some context, you guys are going to be working with this protein language model, ESM. It is an encoder-only model. You're familiar with ChatGPT. ChatGPT is a decoder-only model. Google Translate is an encoder-decoder. And so we can kind of like mix and match like different pieces of this. Now you guys have kind of all the information. And next day, Sarah is going to tell you all about the workings and how we use protein language models. So I'm looking forward to that. All right. So do you know how to stop the Zoom?
Maybe stop sharing first. That
Stop
button.
sharing. Okay.
And then there may be a button that stops recording. Oh, that one, pause stop recording.
Ah.
Yes.